Hello, everybody. Does everybody hear me and see me well? Okay, so it's been about a minute. Perhaps more people will start pouring in throughout the next few minutes. So I'll just get started. Welcome to this educational session number 2. So the educational sessions are made up of 2 talks that last approximately 20 minutes each. And for the first of these talks, today we have a special roundup of lightning talks that have been prepared by this year's OSIG Fellows. So the OSIG Fellows are 10 early career researchers that have been formally recognized by the OSIG. So there we have one whom I will just turn off his mic. So there are 10 early career researchers that have been recognized by the OSIG for their contributions to open science, and they've each prepared 2-minute long videos describing their contributions to open science and or their vision for open science. So half of the fellows were presented at an earlier educational session today, and the other half are going to be presented now. The second talk is titled Building a User-Driven Database of Open Neuroscience Projects, and it's given by Matthias Andina. So throughout the talks, please feel free to pose questions in the chat. At the end of the talks, any of the speakers that will have joined, I will invite them on stage and they can address your questions at that time point. And I just want to mention that one of our speakers, Matthias Andina, has mentioned that he will not be able to join the Q&A, but that he's happy to answer any questions through Twitter. So I will post his Twitter on the chat. And finally, if there are any connection issues, you can also view these talks on YouTube by following the link on the green banner that is at the bottom of my video. So if you do that, if you watch through YouTube, just remember at the very end of the talks to join us here again if you have any questions for the speakers. So enjoy the talks. I will get started right now.
Hello, my name is Sin Kim and I'm an Open Science SIG Fellow and a PhD student at Korea Advanced Institute of Science and Technology. Here I'm going to talk about how you too can contribute to open science and make a difference based on my personal experience. There's a saying that every journey starts with a single step, and for even famous open source contributors, that first step could be something as simple as a single typo fix. Well, I'm not famous, but to give a bit of background, my bachelor's degree was in computer science, and what I did as undergraduate involved absolutely no neuroscience. However, I was an active user and occasional contributor to open source, and like many coders reliant on Stack Overflow, I heavily benefit from the open culture in programming. So when I first had to analyze fMRI data, I didn't know where to even begin. Well, I did try asking a few other students who work with fMRI data, but I was not satisfied with their answers because they treated fMRI software like a black box, where you chuck data in and it just happens to work, usually. They would tell me to reference so-and-so papers, but when I looked up the fMRI literature, I found myself even more lost because there were so many small differences between different papers. During all this mess, I came across fMRI Prep. As a programmer, I quickly found myself at home with fMRI Prep, since it was an open source software that shared the same values I did. It was robust, it was easy to use, and most importantly, it was a glass box that anyone can look into and understand. Furthermore, it operated on data organized in BIDS, which is a standard machine-readable data structure that once again made me feel at home. Well, and as a plus, there were several BIDS apps that helped me explore my data in various ways. Well, fMRI didn't exactly work perfectly at first. fMRI had some issues with susceptibility distortion correction, or SDC, for the type of dataset that I was working with. This may be a small step in the pipeline, but from what I could tell, more and more papers consider SDC a necessity, so I cannot ignore this step. So what I did was I posted the issue, and since I needed this myself, I dug into the documentations for the SDC code and software to identify the problem myself. even after all that, I still didn't feel I fully understood the problem, so I read the original methods paper and discussed directly with the fMRI prep maintainers. And well, to be honest, the solution was simple, but in the process, I learned a lot about fMRI data processing and felt a huge sense of accomplishment. This then motivated me to take part in 2020 BrainHack and Open Science Room, and now I'm trying to convince people around me that neuroscience is the hot thing and they too should adopt the practices. For example, collaborating through GitHub rather than emails and Kakao. Our lab also plans to share some new data with open source software like XNAT soon. Finally, I've highlighted the growing recognition of reproducibility crisis through journal clubs. Though small, I hope my actions will help the growing open science scene in Korea.
Hi everyone, my name is Sid Chopra and I'm a PhD student at Monash University in Melbourne, Australia, where I'm using multimodal MRI-based techniques to study brain changes caused by psychiatric medications and disorders. And I want to thank the open science community at OHBM for allowing me to overview some of the contributions I've made to open science. And one thing I'm particularly passionate about is reproducible figures for both data quality and results interpretation and making tools to facilitate this. In neuroscience, and in particular in brain imaging, visualization and figures are an important final product and often form the crux of interpretation. Often these visualizations are made by opening up an image viewer such as FSLI's, ITK Snap, Freeview, or some MATLAB toolbox, and displaying these results and tinkering with the settings until our desired pretty picture is reached. And we can take a screenshot and insert it into our document. For visualizations that involve network and edges, sometimes people are even forced to draw these connections in PowerPoint or some other tool. This process I've just described is at odds with the philosophy of reproducibility. As brain visualizations are often the final product of analysis, they should be generated programmatically using code so that they are reproducible and can be directly compared between studies and analyses. The contribution I want to show you today is an R package I made that allows for visualization of brain connectivity data. While there are some independent packages and software which allow programmatic and code-based generation of figures, we need tools that are easy to use, highly flexible, and integrated into commonly used open source coding languages such as Python and R. The R package I designed is called BrainCon and allows users with a single line of code to generate reproducible 2D and 3D interactive macro-scale brain connectivity diagrams. And I'm going to switch to this and take you through a vignette which outlines the package. So you can download BrainCon by either Googling BrainCon or going to my Git repository, which is SidChop. So after you've loaded the BrainCon package and an example unweighted, undirected connectivity matrix, although these matrices can be directed or weighted, and these examples come with the package, using a line of code, identifying the atlas you use, and there's many preloaded atlases, although it's fairly simple to load your own, and some metrics such as node size. This is the direct output from that line of code, where you can see the edges and nodes displayed on a brain. And there's multiple views and options possible, and this vignette only scratches the surface of the configurations that are possible with BrainCon. Here, the edge weight and node color and view have been modified. And if you're using a weighted matrix, you can change features such as edge weight and edge thickness, as well as the color of the edges, in this case, weighted by connectivity strength. And if you're using directed analyses, such as in DCM or other directed models, arrows are also easily possible. And the same thing here with a weighted directed matrix. And there's some experimental features such as automatic node labeling, although this doesn't work as well if you have a lot of nodes, sorry. And there's some more advanced features such as modulating the size of the nodes based on a feature you like, such as degree in this case, or nodal degree. But often with these figures, while they look pretty and nice and are reproducible, some of the anatomical specificity is lost. So I've also made a BrainCon 3D function, which is the exact same code as previous, but just adding a 3D to the end of the function. And that generates an interactive 3D version of the image. which allows you to explore it, but also take screenshots at set views. And the same parameters can be changed with the 3D image as well. And these can be embedded into your paper. So thanks very much. I hope you find this tool useful. And again, you can reach it by Googling BrainCon and this, or looking at the Git repository for Sid Chop, which is me. And there's some other tools there you might find useful as well. Thank you.
Hello, I am Senior, a PhD candidate at the University of Melbourne. It is an honor to be part of the BrainHack 2021 as an Open Science Fellow for the OHBM Open Science Special Interest Group. To give you a little introduction about myself and my interest in open science practices, I should say that I study different computational methods of modeling brain connectivity. So in my day-to-day research, I use the Python programming language to conduct my research. I use packages such as NumPy and SciPy for data wrangling, Scalearn for machine learning, and Matplotlib for data visualization. I also use more specific neuroimaging packages such as NiBabble whenever needed. In order to make my code publicly accessible to the community, I try my best to apply and implement best practices in reproducibility. I use Git for code version management and publish my code on GitHub. I also use IPython Notebook in combination with Binder in order to make my research more reproducible. In general, I believe implementing open science best practices can generally improve the research quality in the neuroimaging community. That is why I am dedicated to both implementing and promoting such best practices for open science. This year, I am delighted to be part of the BrainHack 2021 as a train track tutor. I will mainly be tutoring for the hands-on sessions of machine learning and data visualization, where we'll be using Python and its packages for machine learning, model fitting, and data visualization. I'm also going to partake in the live Q&A sessions of the same subject on machine learning and data visualization. I hope that you'll find this year's BrainHack interesting, and I'm looking forward to chatting more during the live sessions. Thank you very much for listening.
Hi, my name is Jubesh Ramdani, and I am a second-year PhD student at Trinity College Dublin under the supervision of Dr. Claire Kelly. I'm very honored to be selected as one of the fellows for the OHBM Train Track Program 2021. So today I'm just going to show you how I got started in the whole open science field. So probably the very first thing is, as we all know, that in science, there's no miracles. So, and when we look at papers to try to understand what the authors have done, and when we try to replicate these results, it's not very often that the researchers have explicitly detailed what they have done to get these results. So I got started with open science when I saw that there are so many problems at different stages when we do research, and these problems can manifest at the very beginning when we are going to basically define our own hypotheses, to designing our study, and even when we interpret data and publish our results. So if you're going to attend the Train Track program, especially in the community building session, the one that I will co-lead, I am going to discuss more about these problems that altogether contribute to the reproducibility crisis, as we all know. So I attended a workshop in the UK in 2020. And after that, I got so motivated in starting a reproducibility general club in Dublin, which is the first in Dublin. And the whole reproducibility journal club was started at the University of Oxford. And today we have 133 institutions in 25 different countries. So as the purpose for the journal club, so we all meet and we discuss about the barriers that could impact reproducibility in research, and also talk about potential solutions that could help us counter these effects. So here is an example of the reproducibility workshop in the UK. And as you can see, there are a number of people from different universities, and we all got together from different backgrounds to talk about what we can do to minimize the problems regarding reproducibility crisis. So so for our own journal club in Trinity, we do meet every 2 weeks, but now because of COVID, it's online. So so these are the different examples. When we meet, we discuss, we brainstorm, and very often we have students, undergraduate and postgraduates, and even faculty members and PIs from the broad spectrum of neuroscience to discuss about how we can shift in terms of culture, so how we can incorporate reproducible science in our own work. So I was very lucky, and I got so motivated because I attended the workshop at the very beginning in my PhD. So so my contributions have been to basically do my entire PhD in a reproducible way. So from using openly available data sets to teaching undergraduates about reproducible practices using Google Colab, Jupyter Notebook, and R Markdown, and to teach them and myself how to do reproducible analyses and write codes in an openly and reproducible way, so that later on we can publish all the scripts to help the scientific community. So thank you very much for listening. And if you have any questions, feel free to attend the Trend Track program, or you can reach out to me on email and on Twitter. Thank you.
Hello, my name is Sin Kim, and I'm an Open Science SIG Fellow and a PhD student.
Hello, my name is Matias Andina. I'm originally from Argentina, where I studied biology, and I work as a neuroscientist in a mouse lab. Here are my social account and my GitHub account. You can reach out in social if you want to chat, or you can see some of the projects I work in my GitHub. Today, I'm here to talk about one of these projects, which is Open Neuroscience. What is Open Neuroscience? Open Neuroscience is a user-driven database of neuroscience projects. And I'm going to leave the definition there for a minute and tell a personal story. So the story is the following. I was in my lab thinking about some idea that I just had, and I realized quickly that I had a problem with the tools that I had, and I couldn't make this idea happen because I didn't have the tools available. So initially I was quite sad, and then I remember I'm quite stubborn, so I went on a search, and my thinking at the time was, if only there was a database with open neuroscience projects that I could search and find the solution to my problem. And that's how I found openneuroscience.com. And not only I found the solution for my problem, I also found a team that was open for me to collaborate with, and I joined them, and here we are trying to reach out to you guys about this database of open neuroscience projects. So to sum up, this matters because scientists are trying to do science, and to do science they need tools, and tools are often a major problem. It's either they are very expensive or unreliable or difficult to modify, or they don't talk well between different tools of different vendors. So in general, tools in neuroscience is a major problem. So here I'm showing here an example, and this example is not to make any kind of war between languages or any of the sort. It's just an example of a tool like MATLAB, which is not open and quite expensive. And the alternative tool can be Python, which is open source and free. And in fact, this is an example of a software alternative that includes changing the language, but in fact, you don't have to change the language. There's also an open source alternative in the same or almost the same language. So as the title says here, there's almost always an open solution. And software is easy. You might say there's a lot of community around software and there's a lot of history of open source projects that are successful and broadly used. But what about hardware? Hardware is difficult. It presents a lot of other problems that software doesn't have to deal with. But the truth is that in our database, we have over 200 projects, and most of them are also hardware projects. So we have centrifuge, a centrifuge project. We have a tiny camera to look the inner organs of zebrafish and Drosophila larvae. We have a device to make electrophoresis. We have a 3D printed pellet dispenser for mice that I currently use in my projects. We have, as I said, a lot of projects, and most of them actually are not only software or only hardware. Most of them are both open source hardware and software. So what is open neuroscience? Open neuroscience is a user-driven database of open neuroscience projects. And we focus here in user-driven because our users are the ones generating the content on our website. So let's say a creator has a tool that they want to share with everyone, and they can go on our website and they can access a form, a Google form, and very quickly complete different fields of this form. And we focus our efforts on the back end, where we have some R scripts running, and we are able to generate and host a website. So we basically transform the information that the creator gives us into an HTML file that can be rendered on a normal web browser again. And to promote this content, we use Raspberry Pi hosting a Twitter bot that runs on Python to amplify the posts that we made and some other information that we think is valuable for the community. So for those of you who want to dig a little bit deeper, I promise to be quick on this, but as I said, it's just a text-based pipeline. So we go from something that looks like a CSV file into a markdown file that gets rendered into an HTML file that people see on the phone or a computer, just a normal website. And everything is running on the open. That means that if you go to our GitHub, you can see all the files that we use to generate our site, and you can see the issues that we have, and obviously you can interact with us there. That means that, as I said, if anyone can create or file issues or even create pull requests that we can evaluate to see whether we can change something on our site or not. And basically, it's just a collaborative effort. Everyone is welcome to add their projects. There's a small asterisk there because, yes, we do check for spam, but basically, everyone is welcome to add their projects. That's the only kind of barrier we put in place just to make sure that we are actually generating content from open neuroscience posts. So as I said before, we use a Twitter bot to drive engagement. So this bot will retweet the mentions of open neuroscience, and it will also tweet directly the posts that we generate. And we also can log into the Twitter user, to just tweet ourselves manually. So it's a half bot, half human Twitter account. So how are we doing? We use Plausible, which is an open source analytics tool, and you can see our metrics there, but we also aggregate them and show them in our site. And in the last 12 months, openneuroscience.com has received more than 200 open source projects and around 10,000 visitors. And most of our visitors come from North America or Europe, but we're still very interested in spreading the word out and bringing people from all over the world. This, we believe, is a truly global effort, and we are very happy to see that most countries in the world have accessed our site. We still, as I said, want to expand our reach everywhere, and we are trying to do different things here and there to actually get to the places where it's most difficult, that also are the places that most need open access to tools in science in general, but in neuroscience that's also the case. So to expand our reach, we created this seminar series. We are collaborating with a worldwide organization. This organization hosts different seminar series in different topics. Our topic is obviously open tools in neuroscience, and we have people showing their tools and their software and their creations every Friday, and we live broadcast it on YouTube. So anyone with a cell phone and an internet connection can view it live, or they can access it later on our YouTube channel. And this is awesome because very often people cannot go to the seminars in the university where X, Y, or Z is presenting their tool, and the use of just the live broadcast can enable many people to just go to these seminars when in fact they couldn't have done it. So I think we are trying to leverage the use of this live broadcasting to get the information from the creators themselves to many of the potential users that could never access that information as in the same kind of content as they can do with YouTube. They can also interact with the speakers, so just basically ask questions, again, as a normal seminar that we can be used to, because we cannot normally go to the seminars in person. So we're quite happy that we are doing this seminar series, and it's helping us grow and, as I said, expand the reach of our network. So how are we doing with this? We in Twitter have around 2,000 followers and over 150,000 tweet views. And for our seminar series that just started, as I said, we have around 100 subscribers and growing. And we have, which I think is very important for us, 2,000 video reproductions. And this seminar series started in March, so we are not even a year in. So we are quite happy with the reach that we had so far with this series. So what's next? Where do we go now? So I think the natural path for us is the growth path, and this is not just growth just for the sake of it. It's because our goal is to increase access to open tools in neuroscience. So to increase access to tools in neuroscience, we have to reach almost everyone that can and might need some help with tools around the world. And the problems that we face with this is that tool adoption depends on fragile chains. So when a creator does something, the sphere of influence of that creator, even nowadays, is somewhat limited. And when we think about different creators, those spheres of influence are often independent and disconnected from each other. So two different people are doing the same kind of development, and not often they talk to each other. And that's because they might not know that some other person is also working on the same problem, and they might not have the way to find the project in time. So there's two people working on the same project, and they mostly don't talk between each other, and their users also not necessarily know each other, at least in their immediate spheres of influence. So we have this long chain from creator to user, and this is a fragile chain if the friends of the creator or the immediate collaborators of the creator don't use the tool or don't help with documentation or don't pass the word around. The final user, the one that has no relationship up at the beginning with the creator, is less likely to actually be involved in using the tool. So we think of ourselves as a hub, that we can work as a hub to facilitate this interaction between different creators that can post their projects on our site, and we can help them reach a much wider audience and much wider user base for their creations. On the contrary side, users know where they have to go to get these tools, and it's easier for everyone, and it's more of a fluid system. So hopefully we can expand this and, as I said, increase access to open tools in neuroscience. And we hope that open tools in general are not the afterthought or the alternative, are just the norm. So what can you do? I wanted to switch gears here and get in contact with you, because I believe that you can do a lot for open science tools, and I believe that you can, for example, start by adding a project. If you have a project of open science tools that can be involved with open neuroscience, you can use this link to add your project to our site, or you can share this link with somebody who has a project that you know. Just using this link is very easy to add posts to our site. You can also help us by solving issues in GitHub. You can file issues there and work on some issues that we have identified that we need help with. And basically, you would be helping us with your time, and we would really appreciate that too. You can be social about open neuroscience. If you, as I said, know somebody who has created a great tool, share our site with them, or share our Twitter feed posts so that we can so you can help us grow our audience. And finally, just be awesome. And by being awesome, I mean adopt open neuroscience practice or open science practices in general in your research. If you are in a position of power where you can lead the way your lab works, you can for sure help to make the environment in a way that the environment thinks about open source practices in the first place. But maybe you are not in a position of power. So you're in a part of a team that is not currently using open source practices. Well, you can still share and help implement open source solutions within your team. Maybe you can share some examples of code that you have, or about a tool that you normally use that is open source, and as I said, share with your team so that we have more people using open source solutions. Finally, you can consider sponsoring an open source project, and this can be directly with funds, or it can also be with your time. As I said before, and as we are doing, we are donating our time because we believe this cause is quite important for the development of not only neuroscience but science in general. So both your funds and your time will be highly appreciated. And if you want to know a little bit more about open source and open hardware in particular, you can visit OHBA to learn more about the way that the organization thinks about how to work with open source standards and open source in regards to hardware. The team that I met is these beautiful people, and I'm really happy to work with them. And we are continuously thinking about ways to change this or that in our site, how we can reach more people, how we can make the tools that we have in our database more available to them. So I have to thank them because I found a nice place of like-minded people where I can work in an environment that fosters open neuroscience tools. With that, I want to thank you, and I'm happy to take any questions that you might have.